press it. Okay, I think we're live. Okay, hello everyone on YouTube. Sounds good, looks good. Yeah.
Okay, so we'll start. Yeah. So we'll start recording. Okay, so hello everyone tuning in. This is the April meeting of the Origin of Life Early Career Network, OoLEN. I'm pleased to welcome Moran Frenkel-Pinter and Sreejith Varma to talk to us today about their work in the Origin of Life field. And we'll start with Moran, who is just opened her lab at the Hebrew University. And yeah, take it away, Moran.
Thank you very much, Amit, and thank you for inviting me to give this seminar, and I'm looking forward to discussions. All right. So everyone can see my slides, right? My presentation. Great. So yeah, my name is Moran Frenkel-Pinter, and I guess I started my lab at the Hebrew University about, I guess, a year and a half ago, 2 years ago. The work that I'm going to present today, the study, is in collaboration with Lauren Williams from Georgia Tech. And it is about evolution of complex chemical mixtures. And I do believe that in order to understand origins of life, we have to not just look at the molecules that are at play, but also at the process and how we see emergent phenomena in complex chemical mixtures that interact. So yeah, this is what we're going to talk about. So as we know, chemical and geological processes on the ancient Earth caused a continuous increase in complexity of organic molecules, ultimately leading to the marvelous DNA, RNA, proteins, membrane-forming amphiphas, and so on. And while a lot of the research in origins of life focuses on, very importantly, on the molecules or oligomers that can form from simple abiotic reactions, what we wanted to do in the study that I'm going to present is really to systematically study the process itself, the process by which molecules undergo evolution or changes over time. And in order to do that, we use the platform of just wet-dry cycling, which I'll briefly, I know we talk about this a lot, but I'll briefly discuss the aspects that I would like to emphasize here. But we use wet-dry cycling as a process to evolve molecules. And what we have observed when we systematically did this for complex mixtures and experimentally designed the system in a way that I'll present in a second, we have looked at really the changes over time in a way that enabled us to look at phenomena that was not described before. So the process that we used to evolve the molecules, we used wet-dry cycling, in which we used variations in the water hydration levels. So the system that we used had building blocks or monomers that could condense and dehydrate or oligomerize through dehydration reactions. So when we dry them, basically these monomers link covalently and form oligomers. And when we put them back in the wet phase, we know that, or the expectation is that once we have oligomers, we know that those oligomers that form assemblies that interact with other molecules, they will be the one that will persist over time because assembled structures hydrolyze or degrade basically in water much more slowly than unstructured samples or oligomers. So the idea was to do this like in the wet-dry cycling, do this again and again. And over time, as we do this, we would expect the buildup of more and more persistent species that persist in water that can hydrolyze them. So this is just the system that we use to evolve the molecules. Which molecules we chose for? So we know now that we can generate a variety of, through dry downs or just drying reactions, we can form a variety of different types of oligomers. Some of them resemble proteins. So we have things that have ester bonds or thioester bonds. We can make amide bonds, and we know to make oligosaccharides in these dry down reactions and different amphiphaths. So the idea is to, or here we use simple building blocks. Some of them are used as building blocks for biopolymers today, and some are not. But the idea was to follow them, that these building blocks can link through condensation dehydration and hydrolyze in water. So we have a recursive process that enables to build and to degrade these polymers. And what we did in this experimental design to really understand the evolution or change over time, this complex mixture, we did something that enables us to look at this from a different point of view. What we did is we increased the complexity of the system by increasing the number of reactants. So for instance, we had different sets of reactants, and these mixtures or sets of reactants were nested in a way that set 1 is a subset of set 2, which is a subset of set 3, which is a subset of set 4, and so on. So we just add more and more reactants and basically saw what happens. And I will focus on one of these sets that, but we saw this, what I'm going to show, we saw this again and again in different systems. But what we did is basically we took these different, mixtures of reactants that could link they are connected, the system is connected, the building blocks can react, the oligomers can degrade in water. We had replicates, so 5 replicates, because we wanted to make sure, we wanted to understand basically the variation in our systems. And we did wet-dry cycling to these different mixtures. In this case that I'm going to show, we had 15 dry-wet cycles. Each cycle of wet-dry was for 2 days, so a total of 30 days for this experiment. And we did this under anaerobic conditions to mimic earlier atmosphere, and also to because we worked with oxygen sensitive molecules. So this is an example of the set that I'm going to focus on. These were the building blocks that we used for this one. And you can see it's pretty diverse. We have a nucleobase, a polyo, we have a hydroxy acid, we have a mercapto acid, sugar, and so on. And what we did here is, as I mentioned, we took these different sets and did 15 dry wet cycling under anaerobic conditions. And what we wanted to do is really follow the change over time of the products and monomers in these reactions. And here I'm going to show you some of the analysis that we did. We followed this change over time with a variety of different analytical techniques, including LC-MS, HPLC, NMR, and so on. And here I'm showing a spectra, for example, of this is a C18 HPLC. This is basically separating the different species based on hydrophobicity. And you can see 3 different spectra, one for time 0. These are the reactants, just what we start with. After 10 days, this is after 5 cycles, and after 30 days, after 15 wet dry cycles. So the first thing is that you can see, and each of these is, we call this a peak, or mostly only one chemical species. And what we saw is that over time, as we expected, we saw more and more new peaks or new chemical species. And I'll just focus here on the areas that are shaded in gray. And you can see here, as we enlarge this, this is kind of relative to the amount of the chemical species over time. So from cycle 0 up to cycle 15, you can see, what you can see is that, or what we observed here is that the system kept changing and changing all the time. This is an example of a species that went up and then down. This is something that had a longer lag time but went up. This building block immediately disappeared, and we saw an appearance of new peaks. And we followed these different species, and we know what most of them are. But the most important thing for us here was that we could integrate the different peaks and really follow their change over time. And what we saw is that the system just kept on changing all the time. We see, that between cycle 1 and 2, there is a big change, but also between cycle 14 and 15. So we were very happy about this. So the system keeps changing, but how can we quantify this? So and we really wanted to be able to kind of put a number and say, how much the system is changing when we have a variety of different HPLC peaks and so on. So my postdoc, Pau Aragonas, wrote this code that basically gives us a measure of how much the system is changing. The code is available. The link is in that. We have a ChemArchive version for this. And the code is really available if that's useful to anyone. It automatically basically identifies peaks from HPLC traces and integrates them and measures the rate of change. In this formula, we are basically looking at how much change for the average change for the different chemical species between or contiguous cycles. And 1 is the complete transformation of all species. 0 means no change. What you can see is that as we just start, and you see here the rate of change over cycle number, and you can see that initially, just the first cycle, there's a huge change between the beginning and what we see in the first cycle, which makes sense. And then there's kind of a convergence into still continuous rate of change. Now, this just implies that the system keeps changing, but we also know that what we've noticed over the 15 cycles is that we see changes in concentrations of different species, but we also saw, right? But we also saw that there are new species that emerge and so on. So it's not just changes in concentration. We do see species that emerge. And another thing that we immediately observed is that we didn't get any explosion. So we predicted, as we saw many times when we do these experiments, that we expected to see a variety of different peaks, different oligomers. And this is a calculation of the number of expected product or number of products over number of reactants. So we expected, of course, and this is the green line right here, that shows the theoretical calculated number for number of products up to trimers. So we have just 2 units, dimers or trimers in the system. And we expected as we add more and more reactants, we're going to get more and more products. But what we saw in our experiments, as I mentioned, we did experiments with different number of reactants, and the reactions or the subsets were nested in each other. And what we saw is that this observed kind of purple line is that as we increase the number of reactants, first of all, we didn't get an explosion, which was surprising. But what was more surprising is that the number of products that we saw as we added more and more reactants was not that different. And that was very puzzling for us. And then we looked at the data and saw something that's helped to explain it. And I'll just say that, this is really something that we saw when we carried out the reaction at very low temperature that was pretty low for what we usually do. That's 45 degrees Celsius. And this is when we saw this trend. When we looked closely, try to understand what's going on, we saw something really interesting, which I'll explain right here. So this is a chromatogram, again, we're seeing different peaks. It doesn't matter now for this discussion what they are, but we're seeing different peaks, different species. And this is the blue trace is what happens when we just react two components. Okay, this is, if this is in black, this is the 9-component system, set 3 that I was discussing before, and we see mostly 11 main product peaks. If we just take 2 components from this mixture, we see these peaks. Now, the peaks that are have these lines in blue here are the ones that they show peaks that were present in the 2-component mixture, but were not present in the 9-component mixture. And we saw this again and again. And this is another example in red. This is for 2 other components, glycolic acid and glycerol. When we reacted those, we saw peaks. Some of them were also in the mixture here, but many of them were only evident when we only had the more simple system or the 2-component system, and we almost didn't see them or did not see them at all, or just almost didn't see them when we had the more complex mixture. So to this phenomena we call combinatorial compression, which is that we do not get explosion of species, but also as we add more and more reactants, we actually see omittance or subtractions of species that existed in the more simple mixtures and we didn't see, or we do not see these products when we have also these components, but add more and more reactants. So there is a very stringent chemical selection. We see this again and again. This is a comparison between a 9-component system that we discussed before. This is the red trace, and the blue trace is for the 25-component system. And again, we see different products that we see in a more simple system, but when we add more and more reactants, we just don't see these. As I mentioned, these experiments were done at 45 degrees Celsius, which is a pretty mild temperature for such reactions. And we wanted to understand the temperature dependence of this, what we call combinatorial compression. In order to do so, we took different mixtures and did the reactions at different temperatures from 25 degrees Celsius to 85. And here you can see how it looks like for a 3-component system, 5-component, 9-component. And what you can see is that as expected, so as we increase the temperature, we do get explosion, right? It's just that as we are going to lower and lower temperatures, this is when we're actually seeing this very, very stringent chemical selection. And to better understand the underlying mechanisms that govern these compression, we now have a ChemArchive paper for this that my postdoc did. So he did computer simulations in order to understand what is causing this combinatorial compression that we're seeing. So both the lack of explosion, but also subtraction of species. And we now understand this very well. Basically, we will see compression, well, first of all, as expected, if we have any thermodynamic dominance, if we have some products that are more stable, then of course we will see them more, but this is at equilibrium. But what we're seeing in this system, this is a kinetically controlled system. So whenever we have species that have lower activation energies, they are going to be the ones that are kinetically dominant in the reaction. And they're going to give us basically this or far from equilibrium, we're going to see this compression or selection because of pathways that have lower activation energies. This is showing kind of the size of the dots is showing the compression. This is the number of products as we increase the dominance of the kinetic dominance or the thermodynamic dominance and increase the connectivity. So how much of the different species can react covalently with each other. And as you can see, kind of in the diagonal line, as we increase the connectivity of the system or the dominance, so the differences in activation energies in this case, we are imposing stronger and stronger selection. So we will see compression of the chemical space. And this, for kinetics, it happens far from equilibrium. For thermodynamics, it happens at equilibrium. And we're able to really also basically predict the results that we got. So here you can see the number of species over as we add more and more reactants. And this is if we have basically all reactions have the same activation energies, we're not going to see any differences kinetically, and we're just going to see everything. But if we have species in the purple trace that have some kinetic dominance, then we're going to see this very stringent selection. And this is just a heat diagram showing how we can, for reactions relevant to what I described in terms of the chemistry, we can either change the reaction time or temperature and predict when are we going to be in a kinetic compression regime or going towards explosion and eventually towards thermodynamic compression. This really helps us to also design future systems in which we want to be in the kinetic compression regime. I mean, thermodynamics equals death in the system. So we're definitely need to be constantly looking for being under a kinetic compression regime, which will have long, very important implications for the history of the system and subsequent processes. Yeah, so just another way to look at it or think about is what happens in this system when we look at really initial stages of oligomerization and degradation, really very initial stages. This is, we are basically selecting for reactions that follow the lowest free energy trajectories. Just very quickly, I'll say that we also observed synchronicity in the system. So we followed different species over time, and we've noticed that there is a similarity of trajectories or changes over dry wet cycles of multiple species that basically are aligned and either go up and go down together in concentration and so on. We did this clustering. So, now we're trying to understand this using simulations. So to summarize, we have developed this platform that enabled us to basically follow and quantify rate of chemical changes. And in this system, what we saw is that, even after 1 month, the system kept on changing. I'll just say we also did this experiment for 3 months, and it still keeps on changing. So we're not done, which is good, but we need to understand how to keep it going all the time, right? We see that new chemical spaces are explored, that molecular subpopulations are synchronized, and that combinatorial explosion is really constrained. And by performing these experiments and evolution of chemical systems, we really want to understand in order to be able to manipulate and predict changes in energy harvesting and transitions between chemical spaces, eventually leading to emergence of new phenomena. As I mentioned, this work is in collaboration with Moran Williams and his students. Pau is my postdoc. These are the funding agencies. Just quickly want to mention that we just had a new center, Origins of Life Center, Israeli-German center, that is now a new center for the Origins of Life. And one last slide, just I wanted to briefly say what are we working on also now, in addition to looking at complex chemical mixture. In my lab, we're asking what is special about the building blocks of life, what were the key steps in polymer evolution, what really drives chemical evolution towards an endless, change or endless change, and can chemical evolution be understood, redirected, and repurposed? And these are my wonderful lab members. These are the funding agencies. And thanks again. Thanks for inviting me. And looking forward also to Sreejith's talk. Thank you.
Thank you, Moran. That was a wonderful talk. If anyone has any question on YouTube or here, you're very welcome to write it in the chat. I'm going to start by asking you one question. You show that when you add more reactants to your solution, you get more and more combinatorial compression. Basically, there are the species that are supposed to be there but are not. I wonder if what is your hypothesis to how that mechanistically happened? Do you think there is unevenness in the kinetics that favor more reactive products than others? Or do you think there are more like higher body interactions, some catalysis, some 3-body, 4-body interactions that kind of like favor maybe some products that much more than others, just based on like non-trivial interactions between the reactants you get there?
Yeah, right. That's a great question. So I do think that, what we see here in terms of that we don't get more and more products, or we see other products, we see disappearance of others, is really because as we add more and more, it's the first thing you said, as we add more and more reactants, we are now opening up the chemical space, and we now have a chemical space in which we have reactions that have even lower activation energies. So it all depends, and of course it's not just one it's a network of interacting molecules. But as we add more and more reactants, if we add reactants that do enable lower activation energies, then we are basically going to see those stringent selection towards those products and not the other ones. And you asked about catalysis. Well, I guess I'll break this down into two answers. The first is I would say that there's an intrinsic catalysis here, because we are using molecules that like this mercapto acid that I mentioned, hydroxy acid, that enable reactions through lower activation energy. But that's just and they enable, for instance, formation of peptide bonds that will not occur under these conditions, but they do occur because of reactions, subsequent exchange reactions. So there's intrinsic catalysis here, but it's not the one that you mentioned, which I think I hope that we will see later on as we form longer and longer oligomers. Then I think that this is when we're going to open up the chemical space again into better and better catalysts that will be also more specific. And then this will form of combinatorial compression. Yeah, so I think that what we're seeing is really selectivity at a very, very, very basic level of very simple oligomerization reactions. But then we should see not just this compression, but of course many other phenomena.
Okay, great. Thank you. David, you have a question?
Yeah, two questions. I will begin the first one. Do you get isomers when you observe this increase or plateau at the end in the diversity of compounds, or you just consider one type of molecule and not the isomers?
Yeah. So here isomers, yes, we can detect them in the HPLC. They will have almost always a different retention time if they have two stereocenters. But the calculated here, the green calculated line is not considering is not really considering branching and different isomers. So it is an underestimation. But in what we have detected and quantified, then we can detect most isomers. So
because HPLC is maybe not enough to see the position isomers or stuff like that, where we need sometimes the MS or other times the GC even for separation.
Yeah, it's just it is it's quite nice that C18 column that we used, if we have two stereocenters, then almost always it separates different isomers, but not always. You're right. So it is but we also did NMR, and then this enabled us to get another glimpse of this space. And we have seen very similar results between HPLC and NMR data.
And second question is about the parameters that influence this. Did you test it, or at least in the numerical model, irradiation or different type of matrices, especially different minerals, because some of them might a lot increase this?
Yeah, definitely. it's definitely something that should be done. We have not tested the effect of radiation or minerals on these reactions, but it should be done. And yeah, I mean, the Cronin group had also, I guess, it was not called combinatorial compression, but it was, for instance, the effect of minerals on the Formos reaction. And there's actually also stringent selection there. It's not one change in the number of reactants, but definitely, minerals, metals and many things that we'll do will compress the system, and we should definitely do that.
Thank you.
And Sreejith, you have a question before we move on to your talk.
It's similar to David's question. So, could it be that when you get these bigger and bigger polymers or bigger molecules forming, they get removed from the solution because of its known polarity or hydrophobicity, that you are left with much less components to react with? That could be a reason for this combinatorial compression? Or have you accounted for that?
Yeah, thanks. So yeah, so the question was, is there an aggregation which accounts for, as we increase the number of reactants, this is why we're not seeing. It does not appear, yeah, we have thought about this. Initially, of course, it was just visually, but we couldn't say visually just by not seeing aggregates, everything is soluble. But it did seem like we are not seeing any aggregates. For instance, when we did NMR, that would account for different protons or carbons when labeling them. So we do not think that in this system, we do not think that there was aggregation, but it could definitely be in other systems, or if we would have waited for longer and longer times, that there will be an aggregation effect, and then of course compression in what we detect. But this does not seem to be the case in this experiment. Yeah, so it's definitely the I guess the design in which we add more and more components that are subset, and we're just selecting for reactions that have lower activation energies. But it's a good, it's definitely a good point, and which we thought about.
Okay, great. So thank you, Moran, for the talk, and we'll move
Thank
on to Sreejith's talk right now. Sreejith will talk to us about metabolism and how to analyze and detect it. So Sreejith, take it away.
Are my slides visible? Okay. So good morning, everyone. My name is Sreejith, and I'm an organic chemist by training. My first flavor of origin of life research was during my doctoral studies at the laboratory of Professor Joseph Moran. There, I investigated prebiotic chemistry, especially the early carbon bond forming reactions. And after my doctoral studies there, I moved to Charité University to work in the laboratory of Professor Marcus Ralsa to learn more about metabolism as well as its analytical techniques from the perspective of a biologist. So I was trained as a chemist and moved to biology. So my talk might have aspects of both, but in case if someone doesn't follow it, please let me know. And since my doctoral thesis, I have slightly moved away from origin of life, but the question of how life originated has always been a question that is close to my heart. So many of my investigations were also with this particular question, as in how this metabolism or metabolic structure evolved. So today I want to share with you some of the findings that I observed during my my research, my postdoctoral research. So my research started with CC bond forming reactions that were believed to be the foundation of our metabolic reaction, metabolism. And this happened almost 4 billion years ago in the Archean eon, where there was where the Earth was devoid of any biomolecules and contained only simple abiotic materials like carbon dioxide, water, nitrogen, etc. This, through a process abiogenesis, created what we see the metabolic reaction network, and subsequently into higher ohmic layers, and the role of catalysis was crucial in this process. And the metal and autocatalysis are one of the main aspects of catalysis that played important role. But then how did this conversion actually happen, the chemical reaction? So to gain insight into that through evolutionary studies like phylogenetic analysis, we have it's been found that carbon can be fixed into carbon in the form of carbon dioxide could be fixed into biomass through mainly 6 different carbon fixing pathways. The biggest that we see modern day is the Calvin-Benson cycle, which is the dark phase of the photosynthesis. And there are citric acid cycle, Butyl-Lundal pathway, hydroxypropanate pathway, hydroxypropanate, hydroxybutyrate bicycle, dicarboxylate hydroxybutyrate bicycle. These are different strategies that various organisms use to capture carbon from carbon in the form of carbon dioxide, mostly from its surroundings, to generate different biomass. And the evolutionary studies have shown that the carbon fixing reactions of the citric acid cycle, or which here is shown as RTCA, the reactions of these chemical transformations were oldest. Another feature of many of these reaction cycles are that they are autocatalytic cycle. So by that, what I mean is that when you start with on this right side in this blue field, in the cyclic reaction network, if you start anywhere on this topology and you go one round across the circle along this pathway, you end up with 2 molecules of the material that you started with. So this generates as a way of multiplying the component that has been put into the system, and therefore is showing a replicative kind of behavior. This is the feature for many of the other for the other 5 fixation pathway, except for one, which is the Butyl-Lundal pathway. This is peculiar in the sense that this is a linear pathway, starts with carbon dioxide and ends in acetate or methane. So this a combination of these 2 pathways was believed to be the earliest under certain conditions that have been identified as a candidate for origin of life, such as those you find in underwater hydrothermal vents, or any situation where you have a high temperature, high pressure carbon dioxide. And through our investigations, we were able to show that if you have carbon dioxide at a high pressure, at a high temperature, metallic iron is able to show transformations of Wood-Lundal pathway. And this, when coupled with zinc and chromium ions, you could get many of the sequence of reactions in the citric acid cycle also in one single pot. So this was still slightly incomplete, as in we were still not able to get many of the steps of citric acid cycle, and this was partly due to the steps that require ATP for their activation. So instead of that, we also in the laboratory we also tried to use highly energetic molecules like glyoxylate and pyruvate, which have a reactive carbonyl species. And when we subject them to this mixture to metallic iron at a high temperature, we were able to get much more, that is about 9 out of 11 intermediates of the citric acid cycle. And not just that, other intermediates in the metabolic network was also observed to be formed in the mixture. So this was when I was finishing my doctoral studies, and I wanted to explore what other aspects or what other aspects of metabolism can be observed non-enzymatically in cells. So this brought me to the laboratory of Mark Salzer, who at the same time was doing non-enzymatic reactions of glycolysis and pentose phosphate pathway. Again, in his laboratory, they observed that when you have sugar, such as glucose 6-phosphate, and when you subject it to metallic iron, it establishes intermediates of the glycolysis and pentose phosphate pathway. But here, the distribution was still challenging to detect, as in the concentrations of product formation was slightly less in terms of what we observed earlier. And therefore, you require a really sensitive mass spectrometry to detect many of the intermediates. But nonetheless, we also we were investigating the next sequence of the glycolysis sequence of conversion of sugar to one of the important intermediates of the nucleoside biosynthesis, which was the pentose phosphate pathway. So this pathway is interesting in biology or to biologists because the conversion of glucose to ribose 5-phosphate is an oxidative reaction, and this oxidative reaction is used to generate NADPH, which then helps the cell, or it is used by the cell as a currency against antioxidant stress or to maintain its reduced environments of the cell. So and since DNA biosynthesis is quite a metabolically demanding step, the cell would be able to generate a good deal of NADPH through this reaction. So this reaction was also shown to be non-enzymatic. So we were able, our question was, do you see this non-enzymatic reaction in any organism that is growing at 75 degrees Celsius? So whatever we were able to, whatever we are seeing in non-enzymatically, what we wanted to see was to observe it in an organism. So this brought us to a thermophilic microbe. This called Sulfolobus acidocalarius. This was isolated in 1972 by Thomas D. Brock, who also fathered the discovery of Thermus aquaticus, which is now quite famous for the Taq polymerase in PCR. But coming to Sulfolobus, this is a thermophilic acidophilic archaea that grows at 75 degrees Celsius and at pH 3. So this was similar to the temperature and acidity that we were using for our non-enzymatic reactions. And this studies have showed that this microbes can be grown in a very highly defined media, which consists of glucose and glutamic acid. with a bit of ammonium and metal salts. And with this highly defined media, you can culture this organism with the highly good control of its growth rate. So with this in mind, and it's a high temperature also makes it more convenient for the screen that we were planning to do. So this with this microbe in our hand, I defined a screen of about 42 chemical species compounds made into 180 different combinations. And these combinations was subjected to sulfolobus, and this from after a particular time period of incubation, I measured its biomass, proteome, and its metabolome from the cell culture supernatant. And the two kinds of sets of compounds that I used was arbitrarily divided into categories as nitrogen-free metabolites that would include sugars and dicarboxylic acids, and the second being nitrogen-containing metabolites like amino acids and nucleic acid. So this represents the three different combinations and the 180 different conditions, and it is not the exhaustive combination of the 42 different metabolites, but only as a portion of it. And the reason being is I will explain in the next slide. But before that, what we subject microbe to such a mixture is you observe it would grow in some conditions, and it would not grow in some conditions, and that is exactly we observe, but with a pinch of salt, as in usually the most of the optical density measurements are carried out with the absorption of light, and sometimes some of these metabolites, they themselves are UV responsive. So the actual growth in presence of some of these metabolites can be quite difficult. So that is one point of issue where one place where you might face issue when you rely only on growth optical density as a sign of its growth or not. But nonetheless, we observe two distinct clustering based on their OD, and this represents the ones where the sulfolobus was thriving or was growing in an expected manner, and the red represents clusters or the conditions where sulfolobus had a very slow growth or hardly any growth. So from here, I wanted to get a more deeper insight into how what is actually going on. So for that, I carried out the proteome measurement. So the different 180 different conditions, the cells were then pelleted, lysed, and prepared for mass spectrometric measurement lysed for the proteome, and measured through mass spectroscopy the proteome of the sulfolobus, and the measured proteome was then analyzed through an in-house processing software called DAN that was developed in our laboratory. And this provided the proteome makeup for each of these 180 different conditions that the sulfolobus was subjected to. And from this 180, once we remove the poor growers and selecting for the good growers, we are left with 88 different combinations. And these 88 different combinations are shown in the right side of this figure. And these represents on average 1,300 proteins per condition. So what we are left with from this 180 different combination screen was good growth of sulfolobus in 88 different combinations with about 1,300 proteins. And these proteins would is what we are going to use as a marker or as a signal for some metabolic activity or a metabolic pathway to exist. To identify a pathway now in this mixture of proteins, one strategy that we applied was protein covariation analysis. So the process is simple. That is, you take proteins in pair, protein A and protein B, and then you look for these the variation of these proteins across the different samples that you have. And for those proteins that behave similarly, that is, if one is A is increasing and B is increasing, then we call it positively covarying, and if it's the other way, it's negatively covarying. So when you do such an analysis of all the proteins measured in sulfolobus across these different conditions, we get mainly two different clustering in the heat map that you generate with the two combination, the paired combination, and this could further be visualized as a cluster of proteins, which I would like to call configuration 1 on the left, for the one cluster, and configuration 2 on the other. This could be visualized as two different strategies that the microbe is using for its survival, or for its propagation, or for its survival when it is encountering a certain metabolite. So under certain metabolite conditions, the sulfolobus is employing one metabolic strategy, and in some other metabolic combination, it is using a second metabolic strategy. And when you look deeply into these different configurations, we see that the purine biosynthesis subcluster is one that is varying significantly, and it is highly enriched in configuration 2 compared to configuration 1. And this configuration 2 comprises of metabolites that are quite favorable for sulfolobus, like glucose and glutamic acid, whereas configuration 1, we find metabolites that are either partly helpful, or that causes sulfolobus that requires sulfolobus high change, a large change in its proteome makeup. So coming to the purine biosynthesis module that we see in the configuration, we decided to look more closely at its chemistry. So this starts with ribose 5-phosphate sugar, and the reaction takes it all the way to the purine component called inosine monophosphate. And what you see here are the reaction with two colored bands, the green and the magenta shown on top and bottom. And the green shows the reactant or the metabolite that it requires for the certain transformation. And at the bottom are the end product, or the final the side product that is generated during this reaction. So when you look at it, the reaction usually mainly requires ATP and three amino acids: glycine, aspartic acid, and glutamate, and also 2 carbon sources: carbon dioxide and formic acid. And using just these simple metabolic inputs, the species is able to generate purine biosynthesis, and which is then forms a basis for the DNA and other genetic material. So in sulfolobus, we were able to see detect all the intermediates of this purine biosynthesis pathway in the configuration 2. And looking closely at these materials that it requires for the purine biosynthesis, you realize that they are mostly direct products of the citric acid cycle. So the metabolic requirement is mostly arising from the anaplerosis and the replenishment of citric acid cycle intermediates, and from there it requires some key metabolites like cofactors to generate these specific substrates. So, for example, if it needs aspartic acid, it's produced from oxaloacetic acid and glutamic acid using the cofactor pyridoxal phosphate. And the same is case with glutamine, which is made from ketoglutarate, but in this case, it does reductive amination with ammonium and NADPH. And yeah, the same can be said about the glycine. So what we observe is that the purine biosynthesis, which is conserved across all the different metabolites that promotes the growth in sulfolobus, requires a highly conserved requires a cofactor that contain a highly conserved chemical moiety, that is a pyridine ring. So one of the conclusion or one of the observation I make from this species is that the production of such the synthesis of these chemical moieties is quite critical to its core metabolism. For its core metabolism, and their biosynthetic genes is what I believe to be how would I say to be more important, or is quite relevant for its metabolisms to function. And at the same time, apart from proteome, we also characterize the metabolome of this 180 different conditions. And two of the interesting ones I want to present here is that case of when the sulfolobus was subjected to ornithine, and other case was when it was subjected to the carboxylic acid, glyoxylic acid. So in case of ornithine, what we were observing is the production of the polyamine called cadaverine. And in case of glyoxylic acid, we were observing the production of the amino acid glycine. In case of this glycine, in sulfolobus, it is believed to be produced from serine through deformylation reaction using a tetrahydrofolate as the intermediate. But however, in this particular case, we observed that the glycine was produced from glyoxylate without the serine intermediate, with just using reductive amination. So the NADPH-based, unknown NADPH-based conversion of glyoxylate to glycine is present in sulfolobus. And in case of the ornithine, the situation was that usually in case of ornithine, what we observed was the production of this 5-carbon polyamine cadaverine and other polyamines like spermidine and spermine. But when you look at the molecule of ornithine, it is a molecule with 5-carbon, but usually the polyamine, how it is formed is through the decarboxylation of the carboxylate of the amino acid group. And therefore, it is from ornithine, when it decarboxylate, it should only produce a 4-carbon polyamine, which is putrescine. And in case of sulfolobus, what is happening is in presence of ornithine, there's an enzyme called ornithine decarboxylase that is upregulated. And this decarboxylase is highly unspecific, or its specificity is too low, that it also accepts the lysine as its co-substrate. And this lysine is, upon decarboxylating, provides you with this 5-carbon cadaverine as the product, which doesn't have any other subsequent uses in sulfolobus, and therefore produce in the supernatant. And this was a serendipitous discovery from our end, but nonetheless demonstrate that the ambiguous ambiguity of reactivity of certain sulfolobus enzymes. And this decarboxylating enzyme has a cofactor requirement of pyridoxal phosphate for its decarboxylation activity. So what I have today for you is bits and pieces of information with regard to the metabolic chemistry and how my observations change from early through my investigation with sulfolobus. So what I observe in working with living species is that the metabolic states are manifesting in response to certain nutrient combinations. So if you have a nutrient A and B, you would get perhaps get one metabolic state, and when the system changes or when the same species faces a different metabolic combination, it would change to a different combination. So the entire structure is not conserved, but rather the states are changing depending on the different nutrient combination. And another aspect is many cofactor dependent reaction that we observe in mesophiles. They tend to disappear when you go to case of sulfolobus. For example, in mesophiles, the DNA biosynthesis involves folate-dependent enzymes to produce one carbon sources, but such cofactor dependence is not seen in sulfolobus at high temperature, either perhaps because the temperature is unfavorable for such cofactor-based reactions, or its metabolic biochemistry has a different structure. And from here, I have one question that I've been asking or sharing or asking my friends within Origin of Life field, and I'm also putting it here, as in how many individuals or species are needed for sustaining a population. As in that case, could you call just one being as a LUCA, or could it be actually a group of organisms that could be a LUCA? Is it a LUCA single entity, or is it a group of entity? Is a question. And yeah, and that brings to the end of my talk. And thank you for. And before that, a bit of acknowledgement. I'm really grateful to these two laboratories who had shaped my intellectual journey. Marcus Rals's laboratory at Charité, and Joseph Moran's laboratory at University of Strasbourg. And I acknowledge the funding agencies, and I thank you for your attention, and thank you for giving me opportunity to present my research in front of you, in front of this lovely audience.
Thank you very much, Sreejith. It's been a really great talk. We're running a little bit out of time, so I'm gonna ask you a question from YouTube, and it's also addressed to Moran as well, if you want to answer. The question is that organisms, cells, chemicals, elements, molecules, they're all made of atoms, and we can consider this sort of like the building blocks or the hardware of life. And we cannot solve the origin of life and its further evolution without understanding sort of like the underlying mechanism or the software of life itself. So what do you think is this like software? Because you present here, there are like configurations that you think are very conserved over time. So it seems like there is something that kind of like you know, kind of like controls the hardware into like very specific states. So what do you think that is?
Maybe do you want to go first, Moran? Or should I go first?
Yeah, I mean, I can go first. Yeah, of course, the software, the hardware are so important, and I guess well, in the previous OoLEN seminar, we heard Michael Wang talking about the different kind of types of selection and functional information. And I think, yeah, there are so many levels of complexity, and even the word complexity is complex. So what are we talking about? Atoms, molecules? I think, you know, as we think about this more and more, there are unifying mechanisms, there are unifying, you know, principles, and there and probably different forms of selection apply at different stages, but at all stages, you know, just that some mechanisms are more dominant than others, different along the path of evolution. And you know, as I mentioned, we talked before about catalysis. I think catalysis was from, and now we also heard about this, you know, autocatalysis. Yeah, catalysis, right, for instance, so important, and it was from the very beginning, and it also evolved, so, and enabled increasing complexity. But I just think, of course, it's very complex question that has a that we don't know all of the answer to, and it's we have to think about this. We have to think about, yeah, the unifying mechanisms, and but also how different dominance, how the dominance of these different mechanisms will apply at different stages and emergence. Well, I don't know if that answered partly the question, but
yeah, I mean, I would also agree with that. It says that yeah, capturing this complexity is what limits us from identifying this software. And yeah, I think more and more we research, we would get a more and more paradigm shift, and that should help us yeah, get over this big complexity hurdle to get yeah, understand it. That's what I could also add to.
Great, thank you. David, I'm giving you the privilege of asking the last question.
Thank you so much. Thank you for the presentation, Sreejith. I had a question about the purine production slide you had. How different conditions did you tested to look at the production? Because in the literature, they tested a lot of different conditions, but we still didn't have the full mechanisms for all purine forms. So did you test different conditions that can happen on origin of life, like in pools or in ocean?
So for at least for Sulfolobus, the purine biosynthesis is it's been known since I think 1990s. Except that there was a discussion about the formate incorporation step. So earlier they were believed that it was a folate dependent reaction. So which was like direct manifestation from the eukaryotic steps. But later on, they found that the folate was not that much. I mean, it's there in Sulfolobus, but there was also another thing called methanopterin, which was the main carbon carrier. But then it was shown that some E. coli strains, you know, that was, I think, in 90s, that they could get, or they don't need these folate dependent reactions if they have sufficient quantities of the formate or carbon dioxide in their cells. So that case, there's yeah, it just get rid of this enzyme altogether and just incorporates it using ATP plus formic acid or ATP plus carbonic acid. And the same, it was later shown that in Sulfolobus too, because of, you know, it's a thermophile and it's in a highly stressful environment. So anything that it can do to reduce the amount of genes and proteins, it would go for that. So it has gotten rid of these folate-dependent reactions of synthesis with just non-enzymatic reaction of ATP plus carbon dioxide or ATP plus formate.
Thank you.
Okay, thank you to our two speakers, and thank you to everyone who tuned in. Hope to see you again next month in our next meeting.
Thank you.
Goodbye,
Have a nice
everyone.
day. Thank you again.
Bye.
bye.